Portal vein, where it comes from? Splenic vein and superior mesenteric vein. The spleen, pancreas and the GIT. Okay. From abdominal aorta, there is splenic artery and mesenteric artery. And from GIT, pancreas and spleen, there is a portal vein. Second, again, the thing is that this blood has a less nutrition, less oxygen, but more nutrients. So for oxygenation, supply the hepatic artery from abdominal aorta to celiac trunk and abdominal aorta and hepatic artery. Two goes inside the liver. The basic structure of liver is a hexagonal structure, honeycomb-like structure. At each corner, there is two inputs: portal vein and hepatic artery, and third one is a bile duct, which is a output structure, and fourth one is also there is a lymphatic. Okay, these four things, and in the central there is a central vein. From portal vein and hepatic artery, blood goes from periphery to center. That is called centripetal flow, and from center to periphery there goes the bile and lymphatic. That called the centrifugal flow. Okay, आलोचना बताने clear? Okay. या okay. बराबर मैंने ख्याल से और पूछते नहीं बोल सकता हूँ कल लोग को दिखती है
सरे जाती है
बराबर. Okay. Now internal structure. Your central vein, and this is sinusoid, and this is hepatocyte. Okay? But this your sinusoids are like polar. Okay? And wide gap in between. Okay? And here are the hepatocyte. And this place is called fish log. Then blood comes from here, and actually from here. Okay? Blood comes from here, base of here, comes here. Okay? And from center out hepatic vein to inferior vena cava. Okay? Very large
छोड़ दा
and here is the individual side. Okay. One different kind of cell is present in the sinusoid. Anybody knows? That is the Kupffer cell. Okay? This is actually the monocytes. They are from monocytes or macrophages. But it is not certainly fixed in the sinusoid. So you can call it the fixed macrophages. Okay? So what happens when the blood comes inside from the spleen? The destruction of RBCs. Wherever the RBCs also or toxins or bacteria, whatever it comes, it will engulf here, from here. So the bacteria toxins does not go to the general circulation. Okay? Because liver is the main gateway from GI to general circulation. Okay? Okay. Then some plasma comes here and then fractions come here, and then most of blood goes here, but some plasma remains here and will convert it into the lymph and will go into hepatic. Okay? Okay. Now, these lymphocytes or these hepatocytes, what is the detailed structure of lymphocytes? I will draw it to hepatocytes. Okay. These are two hepatocytes. In between there is a large nucleus, but as age goes increasing, the nucleus will be increasing. Okay. And this sinusoidal surface also has glass. Okay, this is your sinusoid, and this is your base of this. Okay. Now, because of this villi, there is increase in the surface area, so the transport mechanism will occur easily. There is so many transport mechanism occurs here in this case of usage. Suppose there is amino acid comes inside the hepatocyte, then becomes the plasma protein. Okay, then plasma will again enter into the space of usage, and from there it goes to the blood into the circulation. Again, RBC from the preparation of RBC comes here, bilirubin comes here, bilirubin will uptake from here to here. Okay, bilirubin conjugation of the bilirubin by glucuronic acid. This comes the conjugation of the bilirubin. But when the bilirubin is conjugated, it cannot come out from the hepatocyte on this side. Okay, there is a system in here. Actually, two hepatocytes are like this. Okay. Okay, and this is the biliary canaliculi. Biliary canaliculi not start as a specific structure. It starts as a gap in between the hepatocytes. Okay, so from this starting is a here to a tight junction, and here the bile canaliculi. Again, it is not start as a specific structure, bile canaliculi. It is start as a gap in between the hepatocytes. Okay, then when it goes above, આગળ જશે અને પછી canaliculi બનશે. Okay, from here the bile canaliculi is the bilirubin, conjugated bilirubin. There is no transport mechanism from conjugated bilirubin to come from here to here, but there is only one receptor which is here. So what happens when bilirubin is conjugated? It cannot go outside here, but it goes here and here into the bile canaliculi. From bile canaliculi it goes into the bile duct and to the GI tube. Okay. Again, there are some drugs. When you take the drugs, okay, there must be some modification. It will be activation. Okay. So some drugs come here, hepatocytes stay inside. Then it may be possible that the metabolite goes into the bile and into the GIT and excrete in the fecal matter. There are some drugs which are taken up, metabolized, and then again it will go into the space of tissue, goes to the blood, and it will excrete in the urine. So there will be two types of metabolism. Any drug, whatever drug you take, okay, there will be two excretion, either in the fecal matter or in the urine. Okay, so depending on the drug, it will be metabolized. Okay, Haran Marvel goes on. Okay. Now again, this hepatocyte has many functions. It has many mitochondria, lysosomes, peroxisome, smooth reticulum, endoplasmic reticulum, and rough endoplasmic reticulum. Okay. Okay. What do the smooth endoplasmic reticulum? It produces the lipids, then catabolism of the estrogen, progesterone, testosterone. What do the rough endoplasmic reticulum? It produces the plasma protein. Okay. Catabolism high, production high, secretion high, metabolism high, catabolism type. All things occur in the mostly hepatocytes. Okay. Now again, there is another type of cells which are here. Which space is this? Which is it? Space of this space. This is called stellate cell. Stellate cell, or you can say ito cell. Okay. This stellate cell or ito cells is a fat storage cell. It stores the fat, it stores the vitamin A. Both. Vitamin A is a fat soluble or water soluble? It's a fat soluble. Okay. So it also stores the vitamin A cell. It's very normal, but when some infection occurs in hepatitis, then this cell will throw all the lipids outside and become very aggressive. Okay. So when the hepatitis occurs or any infection occurs, these cells become very dangerous to our liver cells. Okay. When the infection occurs, these ito cells converted into the myofibroblast, and myofibroblast secrete the collagen and fibrin. So your liver will become fibrous liver. It will become the cirrhosis. Again, what happens when this myofibroblast secretes this collagen and fibrin? What happens? This whole will shrink down. This your sinusoid will shrink down. The blood supply will be there less in that area. So what happens in some area there will be reduced blood supply. The hepatocytes and the sinusoid go into necrosis. Okay. In some blood supply, in some area there will be hyper blood supply and the cells will be hyperplastic. So in some area there will be necrosis, some area there will be hyperplastic. So your liver will become nodular liver. Okay. Okay. These hepatocytes. Okay. It will produce plasma protein and phospholipids, and again it produces secret VLDL. What is the name of VLDL? Let's see like a okay. And what is carrying? That is the triglyceride. Again there is the LDL. What is carried? That is the cholesterol. Again there is the HDL. It all together into the hepatocytes, into the liver, from the liver. Okay. Then it also secrete the cholesterol. It also, there is one basic important function of vitamin D. Vitamin D comes into the liver as a cholecalciferol. Right? As a cholecalciferol. Inside the liver, inside the hepatocytes, there is a enzyme that is called 25-hydroxylase. Okay. So the vitamin cholecalciferol becomes the 25-hydroxycholecalciferol. Again, this 25-hydroxycholecalciferol can shade into the kidney. In the kidney, there is a hydroxylase again. There is again hydroxylation at the point of carbon first. So 1,25-dihydroxycholecalciferol. Okay. So in case of the liver disease and in case of the renal disease, there is a severe deficiency of the vitamin D. Okay, vitamin D is the metabolism of both problems. Okay, and again there is a secretion of bile. So there are so many things that liver secretes. Okay. Now, okay, what is bile? Bile નું કમ્પેરિઝન કોઈને ખ્યાલ છે? Bile GIT માં ભણી ગયા છો તમે? Okay, GIT માં bile નથી ભણ્યા? Okay. Bile must be first. First is the bile acid soil, then cholesterol soil. Obviously, water is most of 97 to 98% is the water, but the more solid is the bile acids, then cholesterol, then phospholipids, lecithins. Okay, and IgA, okay, immunoglobulin A. And then bile acids. Which are the bile acids? No. There is a enoxy acid and folic acid and deoxy folic acid. There are two bile acids, okay, into the bile, and again bilirubin. These things are into the bile. Okay. Now, when you look at the liver parenchyma, this is what is called classic structure, hexagonal. This is your classic lobule. Okay. Again, there is one different type that you can describe the liver parenchyma. That is called a total lobule, and the third one is the hepatic okay. Now look at this is your classic lobule. Central vein. Okay. Any problem? Any problem? Yes. Come on. And sit there. Sit there. Please. On third bench. Okay. This is your equator side. This is your classic lobule. Okay. In classic lobule, what happens? Blood comes centrifugal and bile goes centrifugal from center to periphery. Okay. There is one another way to see at the structure as a exocrine function. What we last see is a anatomical structure, hexagonal structure, or you can say endocrine function. Why endocrine function? Because all the things goes into the blood, IgA, bilirubin, all the things. Okay. Second is the exocrine function. What is the exocrine function of the liver? To secrete the bile. Okay. Why it is a exocrine? Because it is not secreted in the blood. Which is secreted into the blood, it is called the endocrine. Which is not secreted in the blood, it is called the exocrine. So bile is a exocrine. Okay. So if I put these three portal central vein, and this triangle, what happens to this triangle? The blood goes from center to periphery. Okay? In portal lobule. This portal lobule is called portal lobule. And bile goes from here. Okay? Your bile duct and the bile from this way. Okay. So again, there is a different kind of portal lobule that you can describe as a exocrine function of the liver. Okay. The bile goes into the portal lobule from the periphery to the center, not in the classical lobule. In classical lobule, bile comes from the center to periphery. In the portal lobule, This is the portal lobule. The bile comes from center to periphery to center. Okay, this is the second type of lobule you can describe. Now there is a third type of lobule. Yeah, कैसे कौन बोली तू? दोस्त ताकत है तो फेस टू फेस बोलिंग बताओ दे, बराबर? वो मजा आ रहा। वो मजा आवे दोस्त ने। जब कोई क्लास में बोली नहीं जाओ। ओके। अगेन
और फिर कुछ ही जान छोड़ा
Hindi na wala ko pa.
central vein, okay? Right. Now these are the sinusoids. Blood goes from here center. Now what happens? These are also connected with the central branches, portal vein and hepatic artery. Now what happens? Here are the hepatocytes, here are the hepatocytes, here are the hepatocytes. What happens? These hepatocytes get the more oxygenation or more nutrition compared to these. Right or wrong? Understand? These hepatocytes get more oxygen, these hepatocytes get less oxygen. Why? Because when the blood goes from here to here, first it goes from hepatocytes and remaining oxygen here and remaining oxygen here. So you can say these are the hepatocytes which get the maximum amount of oxygen. Okay? But as you go from the periphery to center, the oxygen content will be reduced, and these oxygen, these cells will get the less oxygenation as compared to these oxygen, these hepatocytes. Okay? Okay? So these don't get the maximum oxygen and these don't get the less oxygenation. Okay, so according to the perfusion, according to the oxygenation, we can divide the lobule or we can divide the hepatocytes into three zones, 1, 2 and 3. One zone get more oxygenation, second zone get the moderate oxygenation, and this is the third zone which gets the less oxygenation. Again same 1, 2 and 3. Okay, there are three zonations. Okay, why is so much important? Why am These are the sinusoids. There is the first zone gets the more oxidation, there is second zone gets the moderate oxidation, and there is the third zone that is called less oxidation. This is 1, this is 2, and this is 3. Again on the opposite side, the same thing. The zone 1, zone 2, and zone 3. Okay? So these are the areas which get the maximum oxidation, or you can say maximum nutrition. Okay? And these areas get the less oxygen and the less nutrition. Okay? If I put the short here and then long here, then this area from center here to here, then here and here, and that is called the hepatic acina. This is the third type of describing the portal lobule, liver lobule. That is called the hepatic acina. Okay. Now, why it is so much important? Because first we deal with the endocrine function, second deal with the exocrine function, bile, that is portal lobule, and this is the third type of lobule we describe. This is the metabolically function or metabolically or functionally. Okay? Why? In case of congestive cardiac failure, what happens with congestive cardiac failure? There is ischemia. All the organ gets the less blood supply. Okay? Liver, brain, spleen, pancreas, everybody gets the less blood supply. Okay? So what do you think? Which zone affected most in the liver? Zone 3. Okay? Zone 3. Because there is ischemia, there is a less oxygenation. Okay? Here in first zone you can get the oxygen, but as you go from periphery to center, there will be the less oxygen. And because it is a ischemic condition, there is ischemia, there is congestive cardiac failure. By any means there is ischemia. These hepatocytes get less oxygenation, and these hepatocytes will go into the necrosis. Okay? So if there is a congestive cardiac failure, if you take the slide and you see at the hepatocytes or you see at the classic lobules, which portion of the hepatocytes will get necrosed? Zone 3 into the hepatic acinas, or you can say which is this central vein, or you can say pericentral area. Pericentral area of the classic lobule. This is whole classic lobule. This is your portal lobule or hepatic acinas. First, again same here. Again hepatic acinas. Again same here hepatic acinas. So in all case, this is the zone 3. Okay? So in zone 3, all the pericentral hepatocytes will go into the necrosis. Okay? Now, there is a condition of chronic hepatitis. Any infection of the liver. Okay? There is any infection of the liver. So which zone will affect it most? Zone 1, zone 2 or zone 3?
One.
Zone 1. Why? Because if there is infection, there is neutrophils, there are macrophages. It will go into the liver. So which zone is the first one to be exposed to the neutrophils and macrophages? Zone number 1. Okay? Blood goes from here to here. So the zone number 1 will get the macrophages and neutrophils first, then goes to the second zone, then goes to the third zone. So in the case of hepatitis, which zone will suffer most? Zone number 1. Again, there are some toxins. Okay, suppose organophosphate, someone do suicide. Okay, rat poison oil and the pizza. Okay, organophosphate say, some bacteria say, these from goes to the GIT, from GIT goes to the liver. So which zone will affect most? Zone number 1, because these are the zone 1 is the first one to be exposed to the bacteria, toxins or organophosphorus. Okay, and there are also some toxins that carbon tetrachloride or tannic acid or chloroform. Okay, these are the drugs which requires the more oxygenation to detoxify. chloroform, tannic acid, and carbon tetrachloride. These are the drugs or these are the chemicals which requires more oxygenation to detoxify this chemical agent. So which zone will be affected most? 1, 2 or 3? 3. Why? Because in zone 1 you get the more oxygenation. In zone 3 you don't get the more oxygenation. So for detoxification you need the more oxygen. In zone 1 you get the more oxygenation. In 2 there is moderate oxidation, but in 3 there is less oxidation. So when the toxins come from the periphery to center, zone 1 gets the more oxidation, it can detoxify very easily. But in zone 3 it cannot be detoxified very easily. So in that case the zone 3 will get the most affected. Okay. Now I have summarized most of the functions of liver, but in summary, what are the functions of liver? First is the endocrine function. Okay, IgA, bile. No, bile is the exocrine function. But plasma proteins, lipids, amino acids, it will all secreted in the blood. That is called the endocrine function. Bile is exocrine function. Again, there is the metabolic function. Okay, from pancreas, insulin, glucagon will go into the liver, and glucose to glycogen, glycogen to glucose. It will stabilize the blood glucose level in your body. Okay. Then again detoxification, there is some bacteria or toxins goes into the liver, then detoxification also occur into the liver. Okay. Then again there is one specific gluconeogenesis to make the glucose from the non-organic substance. Then called non-carbohydrate source. From there it makes the glucose. That is called the gluconeogenesis. Okay. Now, okay, there is the here suppose this is a bile canaliculi. From the side, this is bile canaliculi. It will come out, then converted into the canal of Herring's. From canal of Herring's it will convert it into the cholangioles, and from cholangioles it will convert it into the hepatic duct. Okay, everybody knows. From right lobe, right hepatic duct, left lobe, left hepatic duct, both converted into the common hepatic duct. Then gallbladder must be of the cystic duct, common hepatic duct and cystic duct combined will make the common bile duct. Then common bile duct will go behind the stomach and goes to the pancreas, then fuse with the pancreatic duct, and this both will open into the second part of the duodenum ample of bladder. Okay. Now, as I said just now, that bile has a water and solid constitution. Okay, the bile secreted into the bile canaliculi from the hepatocyte. Hepatocyte secretes the bile into the bile canaliculi. But as it goes down from the bile canaliculi to gallbladder or common bile duct, it will secrete the inside the water and electrolytes. Okay, once in the gallbladder, when the bile is in the gallbladder, what happens? There are most water absorption from the gallbladder. So what happens? The concentration of bilirubin and bile salts are increasing into the gallbladder. The concentration of bile increasing, and from gallbladder it will go into the GIT. Now bile salt, how it produced? There are two bile acids I just mentioned. Which are the two bile acids? Folic acid and folic acid. Anybody has heard the name of enterohepatic circulation? Yeah, enterohepatic circulation. How it happens? From folic acid and from chenodeoxycholic acid. These are called the primary bile acids. Okay, these are the primary bile acids. The primary bile acids produced in the liver, then it will go into the GIT, into the intestine. Into the intestine, it will convert it into the secondary bile acids. Which are the secondary bile acids? These are the folic acid will occur into the Yeah, deoxycholic and lipocholic. And this one is the lipocholic acid. This is the primary bile acid and this is the secondary bile acid. But this is in the intestine. Okay? So in intestine, it will convert it into the secondary bile acids, and again from the GIT, it will again go into the liver. Okay? Into the liver, it will combine with the glycine and taurine and become the taurocholic and glycocholic acid. Okay? In the liver, it will convert into the taurocholic and into the glycocholic acid, and then it will combine with the sodium or potassium, and sodium taurocholate, potassium taurocholate. Okay, or sodium glycocholate or potassium cholate, and these are called the bile salts. Okay, and what are the functions of bile? Bile का function क्या है? First is the emulsification of fat, reduce the surface tension of the fat, so it can absorb very easily. Okay. Second is the absorption of fat. That two words. Let's go. Cholerectic action. What is cholerectic action? It will stimulate the liver to secrete the bile. Okay, that is called the cholerectic action. And second is the cholegog. Cholegog. What is the cholegog action? It will stimulate the gallbladder. Okay, so it will stimulate the gallbladder. There will be constriction of the gallbladder and release of the bile from the gallbladder. That is called the cholegog action. It will stimulate from the liver to increase the bile, stimulate the gallbladder. And in this mechanism, there is one enzyme or one more that is called cholecystokinin. It will help into the contraction of the gallbladder. Okay? And two things: there is a laxative action and there is a prevention of gallbladder, because bile salt puts out, so there is a precipitation and there will be a gall stone. There is a stone in the gallbladder, but because of the bile salts, there will be prevention of the gallstones. Okay. Now, so in summary, from GIT, all blood goes to the GIT, pancreas and spleen. From spleen, pancreas, GIT goes to the liver. Okay, this is a portal vein. Then again the hepatic artery. Okay, both goes inside into the hexagonal classic lobule. Onto the every corner there is a triad, portal triad, hepatic vein, portal hepatic artery, portal vein, and bile duct. Again there is one is the lymphatic. Into the center there is the central vein. Central vein combined and becomes the interlobular vein, then hepatic vein, and goes into the infra vena cava to the which part of the heart? Right atrium. Okay. From space of Dece, there is sinusoids and there is hepatocytes. There is a space between sinusoids and hepatocytes. That is called the space of Dece. From there, lymph goes into the outside. From hepatocytes, there is a bile canaliculi. Start as a gap in between the hepatocytes. Then comes the canal. First start as a gap. From there, bile goes outside. Okay. Two inputs, two outputs. Not two outputs, but actually three outputs. Okay. Bile duct, lymph, and hepatic vein. And two input, hepatic artery, portal vein. There is a three types of lobules we discussed. First is the classic lobule. Second is the portal lobule. In portal lobule, which is in the center? Portal triad or bile duct. And on the corner, there is a central vein. There is a triangular area that is called the portal lobule. And third one is the hepatic acinus. Okay, in hepatic acinus, long axis, short axis, two central vein and two portal triad. And there is three zones, 1, 2 and 3. One will get the most oxygen, second moderate, and third one is the less oxygen. Okay, there are mostly four types of cells we discussed. Hepatocytes. Okay, mostly hepatocytes is very important. Then sinusoids. Then inside the sinusoid cells, Kupffer cells, we can say macrophage. Okay, and fourth one is the salad cell, hydrocell, and this 4 cell you have to remember. Okay. And then functions of bile and function of liver and discuss. Okay. Any query? Any question? No. I'll take your wisdom.